a lock and triggers a change in the receptor molecule. Signal transduction converts the change in the Cells communicate with one another. The cell targeted by a particular signal and cell response. Reception, transduction, and cell response. The cell targeted by a particular signal has a receptor molecule complementary to the signal molecule or ligand. The ligand fits like a key in a lock and triggers a change in the receptor molecule. Signal transduction converts the change in the receptor to a form that can bring about a cellular response. This might involve a series of steps, a signal transduction pathway that alters and amplifies the change. In the third stage of cell signaling, the transduction process brings about a cellular response. This can be any of many different cellular activities, such as activation of a certain enzyme, rearrangement of the cytoskeleton, or activation of specific genes.
When you look at that figure, what we're going to see is that we're actually going to reduce the things that we've already done. The binding of a signal molecule to a receptor protein embedded in the membrane is no different than a substrate binding to an individual enzyme. And all that kinetics that we did with Vmaxes and Kms, we can apply here to determine how efficient the receptor protein is for a particular signal molecule. Different hormones, slightly different shapes of hormones, different shapes of signal molecules may bind with different efficiencies and lead to a different physiological response. The transduction pathway is basically going to be where we change the shape of the protein. In essence, what we're going to do is go from one that's not active to one that becomes active by changing its conformational shape, allowing it to do something physiological. The hard part of cell communication is learning the names of all the individual molecules, the names of the individual proteins, the enzymes, the signal molecules that work through the process, all the different hormones, all the different cofactors, etc., that may be involved in the individual process. But the mechanism is really quite simple. It's the same all the way through. Signal molecules bind to a specific protein that has an affinity for that particular signal molecule. The video referred to them as ligands. The word signal molecule is just as good. It's going to lead to some kind of change in a transduction pathway. You're going to see in a minute that most of the transduction pathways are kind of the same. What they're going to do is they're going to activate an inactive protein by doing something to it, changing its shape and making it a little more active. And then there's going to be some kind of cellular response. So what I'm going to try and do is look at some examples of this. First, we'll look at it in terms of where this kind of thing occurs. Is it in a local environment within the structure of one organ or within a region of cells within the same individual area? If it is, it's called paracrine signaling, as you can see here, a local regulator molecule or signal molecule. Those that are going to then have a hormone released from one cell travel through the circulatory system to another cell or group of cells somewhere else are going to be long distance and are going to be referred to as endocrine signals. We have a couple of really good examples of this. In this particular case, what we see is that this particular cell is releasing signal molecules and there are receptors in the adjacent cells in the area that are then going to respond to that signal molecule and do something in relationship to that local environment for those groups of cells. On this side, we have exactly the same thing. The last lecture of the semester is going to be about neurophysiology and muscle physiology. In neurophysiology, this is a neuronal cell. This is the presynaptic cell. This is the postsynaptic cell. There's a space between it. An electrical impulse comes in. It causes a vesicle. Remember how we made a vesicle? We crossed them through the Golgi, etc. We removed some of the carbohydrates, etc. We made an exocytotic vesicle that's going to release components. In this case, it's going to release a neurotransmitter that will bind to a receptor that occurs in the postsynaptic cell. It's going to lead to an ionic change across that membrane, leads to an electrical change. It's an impulse that is carried like an active potential along the nervous system, etc. But it is still the signal transduction pathway because you have a molecule, it's a signal molecule binding to a receptor in the target cell. And here you have exactly the same thing, except it travels long distances. You have one group of cells, the adrenal cortex produces aldosterone, moves across to the kidney, etc., and leads to changes in sodium absorption.
2 weeks.
So you have two fundamental
2 weeks and 2 hours.
localities within which the signal molecules will work. Paracrine and endocrine. So we call the endocrine system because it goes long distance. Now I'm going to give you a couple of examples. We'll start with a very simple kind of model in which we're looking at these cells capable of existing in two different genome habitats. It can be a haploid cell or it can be a diploid cell. In the diploid cell, it has twice the amount of normal gametic genetic material, and in this particular case, we have two haploid cells. One is the A cell, one is the alpha cell. The A cell produces a factor called the A factor, which is recognized by a receptor protein on the alpha cell. The alpha cell produces a protein, the A protein, that is recognized by a receptor on the A cell. The two cells get together, they fuse, and you now have a diploid cell. One of the ways in which the genetic apparatus of these cells can function, either as individual haploid cells, happy as can be, etc., or they can mate and fuse together and give you a diploid organism and exchange their genetic material. When we get to mitosis and meiosis, we'll look at mechanisms of how genes are changed and passed from cell to cell through the process. But it is using the signal transduction pathway. Another example. In this particular case, we have cell-to-cell contact. Last week, we looked at the intracellular junctions between cells. In animal cells, we saw that there were gap junctions. Gap junctions are relatively small, okay? Lower than a nanometer in diameter, etc., which will only pass very small molecules. Whereas the plasmodesma that exists between plant cells are quite large. It can literally pass organelles about the size of ribosome, and maybe even something a little bit larger, some small mitochondria, etc., that can go from one cell to another cell. It is a method of communication. It's not the signal transduction pathway, but it is a way in which you can exchange material between cells. So it is a communicative mechanism between the two. Another example is using receptors to bind to signal molecules embedded in the membranes of other cells to bring the two cells into close contact. This turns out to be a very important process in embryological development in a large number of organisms. For example, the lens tissue in your eye develops because of its contact with adjacent cells, okay, which will allow the passage of chemicals between those two individual cells. And that is then going to lead to a differentiation of crystalline protein in the lens of the eye, etc. The developmental biologists often want to know what is regulating and controlling the growth of individual groups of cells during the embryological development of individual organisms. And they've done experiments like this in which you have prevented the presumptive lens tissue from contacting the adjacent tissues by putting a small piece of glass or silica or something like that between the two cells, preventing them from contacting, and then the lens tissue doesn't develop. So it is a kind of signal transduction. It's a recognition, not unlike yeast cells, etc., in which the two cells recognize each other, leads to some kinds of responses in those individual cells. Now, we're going to look at the details of a couple of examples. There are thousands. This is just going to be sort of a generic kind of thing. First, we're going to take a look at the reception process. As I told you before, and I showed you when we were looking at membranes, the ability of things to go by facilitated diffusion. We were looking at the ability of the Michaelis-Menten equation describing the affinity of the signal molecule for its receptor or substrate for its particular enzyme. So you can see that down here. Forming the complex between the two is no different than forming an enzyme substrate complex. So mathematically, you can treat it in exactly the same way. I can determine the efficiency with which a receptor protein, a protein embedded in a postsynaptic cell, recognizes the neurotransmitter that's coming across through the synapse from the nervous system of organisms. Secondly is transduction. Transduction basically is going to change the conformational shape of proteins as they go from an inactive form to an active form. And then the third part is going to be a response. Physiologically, it's going to be. So let's take a look. This is the diagram that I used as a second. No different than we saw before. Except I've added some things to it. What we see here is a signal molecule, maybe it's a hormone like epinephrine, binding to a receptor protein. It's going to lead to some kind of change over here. Now, one of the changes that we're going to look at is the concept of internal signal molecules inside the cell, elicited by an external molecule. In this case, the signal molecule on the outside is going to generate a molecule on the inside that will become another signal molecule. And they end up being called second messengers, because the first messenger is the signal molecule itself, the second messenger is a molecule that's created inside the cell. And one of those molecules turns out to be cyclic AMP. It's critically important in lots of physiological responses in cells. Now, what is cyclic AMP? Well, here's ATP. The A, the sugar molecule, notice it's the ribose form of the sugar, etc., and the 3-phosphate. There's an enzyme called adenyl cyclase, which is going to break off the bond at this point and remove the 2 phosphates. And then it's going to allow the phosphate to cycle back through the number 5 carbon itself and form a little ring structure. Well, it's called cyclic AMP because it only has 1 phosphate. It's adenosine monophosphate, AMP, but it occurs in a cyclic fashion. And it turns out to be a second messenger molecule. That means it's going to do something else for things inside the cell. Now, one of the reasons I want to show you this at this particular point is because I want to talk about the gentleman who discovered it. His name was Earl Sutherland. He was a biochemist at Vanderbilt University who in the 1950s was able to determine what the role of epinephrine was in glycogen metabolism in animal cell systems, etc., by discovering the molecule cyclic AMP, which elicited the breakdown of glycogen in cells. In 1971, they gave him the Nobel Prize. He's a very interesting gentleman. He was a pretty good athlete in high school. He was always active, and he was an avid fisherman. I mean, a really avid fisherman. He owned a big Chris Craft cruiser that you could go out into the Gulf of Mexico or off into the Atlantic and fish for marlin and so on. Now, the reason I mention that has nothing to do with it, is to talk a little bit about how medical schools become famous. Shortly after winning the Nobel Prize, at a time when the University of Miami was sort of maybe transitioning from suntan youth to a much more realistic academic institution. One of the ways you can do that is buy Nobel laureates. And we went out and we bought a Nobel laureate. We made an offer to Earl Sutherland to move to the University of Miami. You click on the link up there, it'll take you to a page that's now in his honor on the UN website. Because he was an avid fisherman and had a home in the Keys. So we told him, hey, it's a shorter trip from Miami to the Keys than it is from Vanderbilt to Tennessee to the Keys. And he bought the deal. He made his name, said and all that kind of stuff. And so he and his entire entourage, some 15 to 20 people, moved from Vanderbilt University to the University of Miami. We've done that a couple of times. If I told you on the first day of class, Shapley was another one that we got from Tulane after Katrina had destroyed Tulane University. So it's one of the ways you do it. Now, unfortunately, for the University of Miami, for 2 years after he got here, he died. Really kind of sad. If you go to the link, what you're going to find out is his entire library of papers that he's published, all his notes and all his procedures, etc., are stored in the University of Miami Medical School library, the Culver Library. I thought, sometimes I think it's interesting to know some of the humanistic nature of some of the discoveries that we're going to look at in this class. So now let's talk about, a specific example. In this particular case, what I'm gonna do is look at what are called G protein receptors. This is a G protein. It has embedded into the phospholipid bilayer a series of alpha helices, et cetera, that anchor into that particular membrane at that point, et cetera. And it has a place at which it can bind a signal molecule on the outside, and on the inside, it has a segment which is going to attach a group of proteins that are called G proteins. What's a G protein? It's a protein that binds and releases GDP and GTP. GTP and GDP are the same as ADP and ATP, except it's the G base rather than the A base in each one of them. That particular receptor protein is going to function in the following way. Here's the G protein coupled receptor. There's the signal molecule it binds to. It changes the shape of the signal molecule so that the signal molecule now attracts what's called a G protein. The G protein that it attracts is the one that is inactive, that has the GDP form of the nucleotide bound to that particular protein. When it interacts now with an activated receptor, okay, it displaces the GDP and replaces it with GTP, which now converts it to an active form of the G protein. And it migrates in the membrane. And it migrates to an enzyme, okay, and activates that enzyme, which will lead to a cellular response. Now, in order not to continue to have a constitutive, continual stimulation of the G protein, you need to get rid of it. So there is a GTP ACE that will break apart the GTP back to GTP and make the G protein inactive. So signal molecule binds to the receptor, converts an inactive protein to an active protein. The active protein is going to do something else down the line. Thousands of different G proteins function in exactly the same manner. And here's a more specific example. In this particular case, okay, we have a signal molecule, epinephrine. Epinephrine is going to bind to a G protein receptor, and that G protein receptor, okay, is going to activate a G protein. And the protein that is going to activate in this case is the one that Sutherland discovered at adenyl cyclase, and so it's going to make cyclic AMP, the second messenger inside the cell. And that second messenger now is going to bind to an inactive form of a kinase enzyme, kinase A, and convert it to an active form. Remember what a kinase is? What does a kinase do? A kinase is a transferase enzyme that's going to transfer a phosphate from ATP on to some of the molecules. In this case, it's going to phosphorylate another protein. And the protein that it's going to phosphorylate is an inactive form that's going to become active. And that active kinase is now going to activate another inactive kinase that's going to activate another kinase, which is going to enact another one, and another one, and another one, and another one, and go on down the line. You have this phosphorylation cascade of constantly converting a series of inactive proteins to active forms. Now, we started here with one signal molecule. We activated several G proteins. We activated several adenyl cyclases, which made lots and lots of cyclic AMP, which made lots of inactives active, lots more of inactives actives, lots more of inactives actives, lots more of inactive actives. So one signal molecule can generate 1,000 activated molecules at the bottom. What does it do? Well, the last thing that gets activated is an enzyme called phosphorylase. It converts an inactive form of phosphorylase by phosphorylating it to an active form of phosphorylase. And what does it do? It hydrolyzes a glucose unit off of phosphoglycogen. So you're going to dump lots and lots and lots of glucose. Okay, starting with one signal molecule of one epinephrine, you made 1 million molecules of the phosphorylase enzyme that are now active, that are all converting glycogen into glucose. And so when the gorilla walks in the room, you can make a decision of whether you want to fight or flight. The classical response kind of system. Okay. This is how you get your energy. Get it again. By the signal transduction mechanism of activating a signal molecule or a phosphorylation cascade that leads to energy production. The significance of this, okay, is that you're activating lots more molecules with a single step at the beginning of the process.
Receptors that are plasma membrane proteins usually affect the cell through multi-step signal transduction pathways. These pathways allow for amplification of signals and signal coordination and regulation. Signal transduction often involves smaller molecules called second messengers and protein phosphorylation by enzymes called protein kinases. Second messengers are small, non-protein molecules that act as intermediaries in signal transmission. Two important second messengers are cyclic AMP and calcium ions.
Cyclic AMP acts as a second messenger in pathways initiated by both G protein-linked receptors and receptor tyrosine kinases.
The hormone epinephrine acts via cyclic AMP as a second messenger. An epinephrine molecule docks with a receptor protein, which acts through a G protein to activate the enzyme adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP, or CAMP, which quickly diffuses through the cell and triggers further steps in the signaling pathway. In this cell, we show it activating a protein kinase. Calcium ions also act as second messengers in signal transduction pathways.
This time we will show a receptor tyrosine kinase, but G protein-linked receptors can also initiate release of calcium ions. The signal molecule docks with receptor and activates an enzyme called phospholipase C. The enzyme cleaves a small molecule called inositol trisphosphate, IP3, from a certain kind of membrane phospholipid. IP3 docks with a calcium channel on the endoplasmic reticulum. The channel opens, releasing calcium ions into the cytoplasm, where they activate proteins that carry out cell responses.
Enzymes called protein kinases are important links in many signal transduction pathways. A protein kinase catalyzes the transfer of phosphate groups from ATP to another protein. More often than not, the activated protein is also a protein kinase, which may act on still another protein kinase. One kinase may activate many molecules of the next type of kinase in the chain, thus amplifying the signal until the last kinase activates many molecules of the protein that carries out the final cellular response.
One thing that the video did not tell you is it said that calcium and cyclic AMP were signal molecules. Well, the inositol 3-phosphate itself is also a signal molecule released from the phospholipid in the membrane. This becomes the signal molecule that activates the calcium ion channel, which will open up and allow calcium ions to diffuse from an area to higher concentration to lower concentration. When we look at muscle contraction, what we're going to see is that the key signal molecule during relative muscle contraction turns out to be ions of calcium released by the endoplasmic reticulum in cellular muscle. Now, I don't want you to think that it's as simple as it is, because I want you to look at a couple of other examples in which we have a multiplicity of responses. Here we have an example that's entirely different. We have a signal molecule. Here's the hormone aldosterone, released by the adrenal gland, sitting on top of the kidney, etc. And what do? They transport across the membrane to an internal receptor protein. That receptor protein has an affinity to go through the nuclear pore. Remember, it was difficult to get things through the nuclear pore unless they had a nuclear localizing signal as part of the structure of that individual complex, etc. And it gets inside and it becomes an active transcription factor which will bind to a particular region of DNA, synthesize a new series of enzymes, etc., which will then move out of the nucleus into the cytosol, and they're going to make a number of enzymes which will regulate sodium and water flow in the kidney. So here you have a hormone that's going to elicit proteins that's going to regulate osmotic balances within the kidney. using the signal transduction mechanism. Here's another example. This is the one that I've been talking about in terms of neurophysiology extent. You have a presynaptic cell, you have a postsynaptic cell, and you have the diffusive space between the two of them in which a neurotransmitter, okay, is going to be released into that space and diffuse across that. So here we have the synaptic vesicles, okay, they're going to bind, they're going to release the neurotransmitter, and they're going to bind to a receptor on this side. Here's an example of a receptor. This is a gated ion channel. That means it can be open or closed depending upon whether or not specific signal molecules are bound to it. In this particular case, the neurotransmitter binds to that particular receptor and opens the channel, allowing ions to flow across that channel into the postsynaptic cell. What does that mean? Well, since it's an ion, it's going to move electrical charge. If it's sufficient, if it's above the threshold level, changing the electrical charge in the postsynaptic cell, the one on this side, it may elicit an action potential, and you end up with a neurophysiological response to whatever the original signal was that came in on this side. So again, it is the signal transduction process. Now, when you look at all the nerves and all the different neurotransmitters, etc., that are the individual signal molecules, you get the idea of the complexity of the system. Whereas the concept of how it works, again, is somewhat simplistic in its design. The third is we're going to activate with growth factors, things that will stimulate cell division using the signal molecules again. In this particular case, the growth factor is something that binds to a particular receptor in the membrane, and it leads to a phosphorylation cascade that results in an active protein moving through the nuclear pore to activate an inactive transcription factor to an active transcription factor, activates particular genes. Those genes may be the ones that initiate the process of cell division during mitosis, or meiosis in the cell will eventually divide. So, well, the idea is a little bit simple. The hard part is when you get to upper division courses, you're going to have to go through thousands, I mean literally thousands of molecules that can be activated by cyclic AMP. And then it gets a little more difficult when you need to know the terminology of all this. But again, the concept evolved relatively early and has been pretty stable in terms of the conceptual mechanism, again and again and again with the process. So let's take a breath. Take a breath for a minute, relax, etc. And now we'll move on to cellular metabolism. How cells transform energy, field of bioenergetics, equilibrium, thermodynamics, whatever you want to call it, etc. What we're interested in is how a metabolic pathway works. How does A convert to B? And when B's been produced, how does B convert to C, and so on down the line, in terms of the energy mechanisms that are involved. The kinds of reactions that have to take place in order for a sequence of changes in the series of molecules to go from one substrate to the final product for that individual metabolic pathway. We're going to classify metabolism in a couple of ways. The easiest way is by what they overall do. It's anabolic. That is, it makes small inorganic elements and it makes large molecules. Carbon dioxide and water in the process of photosynthesis, okay, is an anabolic mechanism. You've heard the word anabolic steroids. It's the steroid that stimulates muscle cell growth by stimulating microsatellite cells within the muscle, producing the muscle cells along the side. This is the kind of example that you have. We're going to take carbon dioxide and water, and we're going to make glucose out of it. Now, the opposite of that are organisms that are catabolic. That is, they're going to break down large things into small stuff. So we're going to take organic molecules and we're going to oxidize them down to carbon dioxide and water. This is going to use light energy and ATP and NADPH made in those processes. And this is then going to make ATP that will allow most of the cellular work to occur within the cell. So what we're going to do first is cellular respiration, or catabolic cellular metabolism. Then we'll do photosynthesis. So let's sort of look at the mechanisms of catabolic processes. We can describe those 5 things that you see there. The digestion of the large polymeric molecules, the polysaccharides, the nucleic acids, the proteins that you take in dietarily. As a heterotrophic organism, you're going to catalyze catabolic reactions, breaking those down by hydrolysis in the gut, and then feeding the monomers to a series of metabolic pathways. What we're more interested in than 1 are 2, 3, 4, and 5. The pathways that will convert large organic carbohydrate molecules, glucose, into carbon dioxide and water. Glycolysis, which is going to convert glucose to pyruvate anaerobically without the need for oxygen. We're going to look at the Krebs cycle, which is aerobic oxidation of an intermediate made from pyruvate called acetyl coenzyme A. It's going to break down the carbon dioxide in the water, and it's going to produce reduced pyridine nucleotide, NADH, in the process. Then there's the electron transfer chain, which is going to take the electrons. Where's the energy? The energy is in the covalent bonds in glucose. You take the electrons out of those covalent bonds and you put them on this molecule, and then you take them out of that molecule and you put them on oxygen to make water. But in that process, you create a battery. Across the mitochondrial membrane, you create a proton gradient. The pH difference outside the mitochondria is lower than inside the mitochondria, which favors the diffusion of protons back across mitochondrial membranes. If they do that, they're going to go through an enzyme called ATP synthase, and it's going to link inorganic phosphate to ADP to make ATP. So in words, it's kind of simple. When you look at the metabolic pathways and all the intermediates that are involved, that's when it gets complex. There's 10 steps in glycolysis, there's 10 steps in the Krebs cycle, etc. There's multiple steps in the electron transfer chain, okay, in which we're going to shuttle the electrons through all of them. Keep in the back of your mind, what we're doing is we're oxidizing a substrate. We're stripping the electrons off of glucose, and we're going to put those into a carrier molecule, the reduced form of NADH. We're going to take the electrons off of that, we're going to add them on to oxygen to make water. In the process, we're going to create a battery across the cell membrane. Difference in charge across the inner mitochondrial membrane. That's what we're going to do. We're going to look at all those individual steps. And as we go, I'll tell you what I think is important for you to remember in the process. And then again, what is not important in the process. Now to understand this, we need to understand how cells transform energy from one form to another. And there are the laws of equilibrium thermodynamics that describe how energy transformations take place in the physical world as well as in the biological world. The concept of energy is theoretically the ability, okay, to do some kind of work, and in that process to release heat.
Energy is the capacity to cause change. Kinetic energy is the energy associated with motion. Energy that is not kinetic is called potential energy, and is energy that matter possesses because of its location or structure. Energy is neither created nor destroyed, but converted from one form to another. This property is called the conservation of energy.
The bow and arrow start out with low potential energy. As the archer's arm pulls back the string, kinetic energy from the arm's motion is converted to potential energy in the tense bowstring and arrow. When the bowstring is released, the stored potential energy is converted into the kinetic energy of the moving arrow. When the arrow hits the target, its motion ceases. If energy is neither created nor destroyed, where did the energy of the flying arrow go? Let's rewind and look at kinetic energy in more detail. When energy is converted from potential energy to kinetic energy, some of the energy can be used to do work, but some energy ends up as heat, a type of kinetic energy. Heat is the random motion of atoms and molecules. As the arrow flies through the air, heat is generated by friction between the arrow and air molecules. When the arrow strikes the target, all the energy becomes heat energy. The heat energy is rapidly transferred into the air and spreads out. Heat energy is a very disordered kind of energy. It has the highest amount of entropy, or disorder, of any kind of energy. Where do our muscles get energy to perform work, such as pulling back a bowstring? Our bodies use the chemical energy from food to perform work. Chemical energy is a form of potential energy. When your body breaks down food molecules, the stored potential energy from food can be converted to kinetic energy. The stored chemical energy in food is released in your muscle cells during the process of cellular respiration. Using oxygen, cellular respiration converts chemical energy from food to another form of chemical energy called ATP. Water, H2O, and carbon dioxide, CO2, are byproducts of cellular respiration. The potential energy of ATP can be converted to kinetic energy. Some of the kinetic energy is used to do useful work, such as pulling back a bowstring, but the heat energy that is generated cannot be used to do work.
So, where's the energy? The energy is in the covalent bonds of the glucose molecule. The force that holds the two atoms together, the carbons and the hydrogens and the carbons, etc., is where the energy is. And when we break those bonds, we release some of that energy. And some of it is released as heat. But some of it may be captured into other molecules. Overall, we have to lose energy in that process, otherwise it's not going to occur. Now, what do you do with all this energy? Well, the simple answer is that you maintain a constant condition far from equilibrium. Here's an example. We have a fish. The fish either is a seawater fish or it's a freshwater fish. Or maybe it's a tarpon. Like you ever see the tarpon out in the lake? Sometimes you see the fish roll out there. Those are tarpons. They're intertidal. They can go between the two. They need to maintain a constant internal balance of water, whether they're in seawater or whether they're in freshwater. And the two fish respond physiologically differently to those two systems. Okay? So the freshwater fish, okay, is going to drink a lot. The seawater fish is not going to drink a whole lot. There's a big difference in how you respond to that. But the energy to maintain the constant balance osmotically inside the fish, regardless of what the external environment is what we call homeostasis. It's how you maintain a constant pH, okay, in your blood and the like. That's where the vast majority of the energy is made. It's not for movement or any of the other kind of things, but it's to maintain a condition that is far away from equilibrium. And as long as you can do that, you're going to remain alive. So, let's talk about the laws of energetics. Equilibrium, thermodynamics. The first law is energy can either be created or destroyed. At least that's what the physicists tell us. And so how do you determine that? Well, you measure the amount of energy that's there. And you see that you can transform it from one form to another, but you're still dealing with the same amount of energy. And you've done that. How much energy is there in a glucose molecule? Well, you use what's called a bomb calorimeter. All those numbers you see in the grocery store on the packages, 30 calories, 100 calories, less than 100 calories, whatever it happens to be, have been measured, okay, in the laboratories of the food companies, etc., by what's called a bomb calorimeter. We have a water vessel, etc. We have a chamber inside the water vessel in which a sample is put, glucose, etc. We have an ignition box that's going to pass an electric current through it, it's going to burn it, etc. And we're going to measure the temperature of the water chamber. And a calorie is defined as that amount of heat that's necessary to change the temperature of water from 14.5 to 15.5 degrees centigrade in the bomb calorimeter. That's what a calorie is. If you see a kilocalorie, that's 1,000 calories. If you see 1 C, that's 1,000 calories, or 1 kilocalorie. It is the unit that I'm going to use. In reality, physicists actually prefer to use the term joules. And 1 calorie is equal to 4.184 joules. I grew up learning calories. The textbook is written in calories. Grocery stores are written in calories, etc. So we're going to deal with calories and not joules. Now, I told you early on in the course that I'm not really big on details in terms of numbers, dimensions, and things like that. You don't need to remember the dimensions of the mitochondria or all that. You need to remember the dimensions of the ribosome. 20 nanometers. Reference. I'm gonna have one more reference here. The amount of energy that's contained in a mole of glucose. Turns out to be 686 kilocalories. I'm going to use that as a baseline after I have oxidized glucose and converted it into ATP to determine how efficient cellular respiration is in cells by measuring the amount of energy that is produced in ATP that came from 1 mole of glucose. And it's going to turn out that it's a pretty efficient process. We're going to make somewhere in the neighborhood of about 36 or 38 molecules of ATP by cellular respiration, which will give me an efficiency of somewhere near 40% for the conversion of glucose into ATP in cells. There is no human manufacturing system that's 40%. We're somewhere in the neighborhood, 13, 14, maybe 16% efficient in terms of energy transformation in human manufacturing. The cell is much better. Now, the second law of thermodynamics is what sort of guides everything energetically. It's the law of entropy. And the law basically says that the universe tends to maximize its energy. What is entropy? It's a quasi-measure of the order or orderliness of a system. And the universe is becoming more disordered. At some point, it will become so dilute that it's totally disordered, and there will be no universe anymore. At least that's what the physicists tell us. So, I feel sorry about this, and I apologize to you personally for this, but this is what textbook companies do to describe entropy. They pick on college kids. Not quite sure why. First day of school, you come, mom and dad help you set up your room, you build the loft, you get it all nice and neat, everything looks good, etc. They go away, and you go back to your major habits. And then family day comes, coming in a couple of weeks, I guess, early November, something like that. And so you're going to spend a long time going from disorder back to order. How do you go from disorder back to order? You put energy into the system. How do you go from order to disorder? It doesn't take much energy at all. It's the natural tendency of things to become more disordered. That's the easy understanding of the laws of entropy. The rules of the universe. Cities crumble, stars go supernova, we are all equilibrium. What does that mean? I'm equilibrium. There's no way around it chemically for me, I'm gonna die. I can't get out of that at all. You are. Everyone is going to equilibrize at some point. When you no longer can transform cellular energy to stay away from equilibrium, you're going to move to equilibrium, which means you're going to be dead. So let me ask you a question. I shouldn't do this. I really shouldn't. Why come to class when you know that eventually you're going to die? Hey, it's a valid philosophical question.
What do you say? Oh, you're not.
What are you doing here?
What do you say?
Why do you come to class if you
Oh, that's fair.
What you're doing is informing your mind. You're making your mind more ordered. You're expending energy, dollars, to make yourself more intelligent, to stay away from equilibrium, right? So you can theoretically apply the same kind of quasi-principles to what we do in daily life, if you want to think about it. On a day like today, with the rain out there and everything else, it's sort of a valid question. Why should I go to class today? You get the answer. Now, we have a problem. Because I look at a cell. I look at an egg cell. The egg cell becomes fertilized. Single cell. And it divides, makes 2 cells. 4, 8, 16, 32, 64, on and on and on. It becomes more complex. It looks like it's going against the second law of thermodynamics. It's becoming more structured, more detailed, as it begins to grow and develop. And what we forget about that is we try to look at the cell as an isolated system. It's not an isolated system because it exchanges with its environment. It takes in food from the outside. And so you have to factor in all the energy in the system, outside and the inside, and you still lose energy. But for a short period of time, you can work against the laws of thermodynamics. You can become more structured at the expense of energy being transformed. And that energy comes from the food molecules that we take in. Maximum entropy, the universe is nothing but heat. Now, let's talk about how we're gonna do this in individual reactions that could occur inside a cell. I'm gonna convert glucose to other molecules. How do I know which way it's gonna go? Is it gonna go from glucose to carbon dioxide and water, or is it gonna go from carbon dioxide and water to glucose? I have to sort of figure that out. And John Shear applied the principles of equilibrium thermodynamics and physical chemistry to chemical reactions. And he came up with some equations that will define what the energy content is of the system at any point in time and space, particularly in chemical reactions with physical chemistry. And the equation turned out to be delta G is equal to delta H minus T delta S. The delta means that I'm interested in the change in the initial energy and the final energy. 10 versus 1. Okay? There's a difference between 10 and 1. I don't care if it went 9, 8, 7, 6, 5, 4, 3, 2. All I care about is the beginning and the end. In terms of the total energy of the system. How much was there at the start? How much was there at the end? That energy, free energy, theoretically the ability to do work, to do something, to pull the string bow back, to convert potential into kinetic, is equal to the enthalpy of the system. The enthalpy is a difficult term for physical chemists to define, but in essence, we can break it down for biologists too. It's the heat content of the system, how much heat is in the system and how it changes. And that minus T, which is the absolute temperature, times the change in the entropy of the system, the degree of order or disorder of the system. Now, you function at approximately 37 degrees centigrade best. 98.6 is your body temperature, which means that you're not really a physical chemist, because you're working in a very narrow temperature range. If you want to expand it, we're looking at something like 2 degrees, 4 degrees, all the way to maybe 75, 80 degrees, somewhere in that neighborhood. The internal combustion engine in your car takes vaporized hydrocarbons, gasoline, air, oxygen, and ignites it with a spark when the spark plugs. And it explodes. And it releases massive amounts of heat. And the force of that heat in a sealed chamber can move a piston, which moves the camshaft, which moves the drive shaft, and off you go to class. Okay? That's 3,000 degrees. You don't do that in a cell. So for all practical purposes, the free energy in a cell to be able to do something is going to be directly proportional if the heat content doesn't change to the entropy of the system. So all I have to do is to figure out what the degree of order of the system that I'm looking at, maybe a chemical reaction in another cell is. And I remember that we're going to move towards maximum equilibrium. So here's a chemical reaction, AB. Okay? This is a reaction catalyzed by a particular enzyme that converts glucose 6 to glucose 1-phosphate, transfers the phosphate group from one carbon to another carbon. Which of those two molecules is more organized? Okay? Which one has more entropy? Which one has less entropy? Which one is more disordered? Which one is more ordered? Virtually impossible for me to tell. I can't look at that and tell you which one is more ordered than the other. But this is where Gibbs was very clever. He figured out a way to get around that chemical process by deriving the equation into another form. Delta G is equal to delta H 0 prime plus RT natural law concentration product over reaction. What does all that mean? Well, delta G is the actual free energy at any point in time and space of a reaction. The reaction starts here. I'm going to measure delta G here. Then the next day I'm going to measure it here. So the values will be different as it progresses from substrate all the way to product. What's delta G 0 prime? It's the free energy change that exists under idealized conditions in the laboratory. Usually 1 molar concentration. 1 mole of substrates, 1 mole of products, etc., constant temperature, everything. We set up conditions in the lab to measure what the energy content will be under given circumstances. R is the gas constant, T is the absolute temperature, Ln is the natural law of conversion factor. And we used to have to do that in the old days when they had slide rules and we didn't have calculators to convert it, etc. Now it's just much more simple. So at equilibrium, what is the free energy of any reaction? If the reaction glucose 6 to 1 or glucose 1 to 6, what's the free energy change? Well, by definition, at equilibrium, 0. There is no energy able to do work when you are at equilibrium. You are dead. Okay? So, I can now use that within the equation to determine what the actual standard free energy is at some point. So, delta G is equal to the equation. At equilibrium, delta G is 0, so 0 is equal to delta G 0 plus RT natural log concentration over plot. I'm going to rearrange, subtract the RT side from the other, so I end up with delta G 0 prime. The standard free energy is equal to minus RT natural log products over reactants. At equilibrium, the concentration of products over reactants by definition is called the equilibrium constant. It's what you would expect it to be. No matter what the reaction is, when it goes to equilibrium, it will be that concentration. Now, if I solve it, I'm a biologist. I can simplify all of this. I'm not a physical chemist. I don't have to change the temperature. I don't have to change the pressure. Okay? So I can make all of this very simple. Okay? Delta G minus RT natural log P over R becomes 2 times 298 times 2.03 log to the base 10. So delta G 0 prime is equal to minus 1372 log to the base 10 is the equilibrium constant. Do you see what happened? You see what the value of mathematics is, what gives really finally real life? I couldn't measure delta S. I couldn't measure entropy. There's no entropy in this equation. Okay? What's in that equation? Concentration. I can measure the concentration of a reactant at the beginning and at the end or at equilibrium. And so I've eliminated the need for having to determine a term I cannot determine by replacing it with terms that I can easily measure in the lab, whatever the concentrations are. Now, it actually makes logical sense if you think about it. Let me show you. Look at this relationship. Okay? There's the equation, the standard free energy, minus 1372 log to the base 10 of KEQ. As long as we're a biologist, it will always be that. It won't change. Now let's look at what happens at equilibrium. At equilibrium, I measure the concentration. It turns out that the product is 1 and the reactant is 1,000. What does that tell you? It tells you the reactant doesn't want to go to product, period. Right? If it did, they'd be equal, 500 and 500. So, if I put it in the equation, okay, log to the base 10 becomes minus 3 times minus 1362, so it becomes plus 4116. What's that mean? That means that's the amount of energy I would have to get the reactant to go to product in this circumstance. At the opposite end, I have a lot of product and very little reactant. Exact opposite. If I put the numbers in now, I have plus 3 times a minus becomes a minus 14, a 4116. The product is easily converted to reactant. So what do I have? I have a system in which I can look at and measure whether the free energy change is positive or negative. If it's negative, okay, the reaction is going to occur. Free energy is going to be released. If it's positive, the reaction is not likely to occur unless I add that amount of energy for the system to work. Well, I took something I couldn't measure, and I put it in terms of something I can measure, and now I have a really simple kind of thing. What is the standard free energy? Well, you go to a handbook, chemistry and physics, and you look up standard free energy. Look at all these reactions, all of them. Hey, here's a one. ATP plus water yields ADP plus inorganic process. We've been doing that all along, right? What's the free energy of that? The free energy is negative by a value of 7.3 kilocalories, 7,300 calories per mole. That means that's a favorite thermodynamic reaction. To go in the opposite direction, I would have to add plus 7.3 kilocalories per mole. It's simple. How does metabolism work? As long as it's negative, it's gonna work. If it's positive, it's not gonna occur. Maybe there's a way we can make it. Let's see. Exergonic reactions, those in which the products have less energy than the reactants, they're spontaneous. They're going to occur, period, because they have a negative free energy value. You go camping, you build a fire, okay? You put the marshmallow over the fire, you get the graham cracker and the chocolate, you make some more. What are you doing with the wood? What is wood? Wood is polymers of cellulose. Polymers of glucose, the same as starch, same as amylose, etc. Glycogen, it's all the same. The difference is that here, I'm releasing massive amounts of heat all at once. I'm not conserving any of the energy at all. For cellular respiration, we're going to take it one tiny little step at a time. I'm not going to burn all the covalent bonds at once, I'm going to break it one. Then I'll wait a little while and I'll break another, and then another, and then another. So you get the same amount of energy in both of them. You release 686 kilocalories per mole of glucose, no matter how you cut it. It's just the difference in how you do it. What Cells figured out is a very neat way of doing it slowly, one reaction at a time in that process. It's endergonic. It has a positive free energy value. Here's a good summary. Energy reactant, this is the amount of energy that's available, that's the amount that you get at the end, it has a negative delta G. This is what you start with, this is what you want. You don't have enough energy to do it until you add some energy to the process. Positive delta G in the system. This is how life works. This is how metabolism works. Because in essence, what we're going to do is we're going to link things together that are not fixed with things that are fixed. So that the final energy, overall, in a metabolic pathway, A to Z, will be negative. But there's some steps in between that are positive, that can go. We'll see how. Take a look. Here's another thing you can think about. Life is thermodynamically far from equilibrium. I'm far from equilibrium. It turns out that there may be a way in which we can sort of answer why we stay at equilibrium for a long period of time. And it depends really somewhat simplistically on the concept of the rates of reactions of hydrolysis and condensation. If you look at those two reactions, look at it in this diagram, etc. Here we have a condensation reaction. I'm going to link those two amino acids together with a peptide bond between them, and I'm going to form a peptide down here. Turns out that that reaction is faster than the opposite of that. The hydrolysis. When you take in proteins at lunch, you oxidize all the proteins by hydrolyzing them, taking water across those bonds. That turns out to be slow. So theoretically, you can build things faster than you can take them apart by hydrolysis inside a cell. As long as we can theoretically continue to do that, we can stay away from equilibrium. It's important to remember that life is a condition that is not equilibrium. So, let's look at metabolic pathways. Here's a reaction. Glucose plus fructose 6-phosphate is going to make some sucrose. It's going to make a little bit of sugar. We measure the standard free energy, or we look up the standard free energy of that in the table, etc. We find that the value is plus 5 kilocalories per mole. That's not thermodynamically effective, unless I add that amount of energy to the system. So what am I going to do? I'm going to hydrolyze ATP. How much energy would I release if I hydrolyze ATP? I showed you the number before. That's probably a number you should also commit to memory. 686 kilocalories for glucose, 7.3 kilocalories for the hydrolysis of ATP. So let's do that. Here's ATP. Here's the adenine unit. Here's the ribose sugar. 3 phosphates. We get water across that. We break that phosphate. And the free energy change in that, in terms of energy available to do work, is 7.3 kilocalories, based on concentrations using the equation we did before. Now, something I want you to keep in mind as we go through. We're going to look at metabolism, and most of it is going to be involving ATP. There's a lot of energetic reactions that include the other nucleotides as well. CTP, CTP, TTP, and UTP. All of them. Okay? The only difference between all of these nucleotides is whatever the nitrogenous base is. The energy content is in the front terminal phosphates. The bonds that you see right here, these are high energy phosphorylated bonds that give up more energy than you would expect them to do, and that's why they're called high energy. So it really makes absolutely no difference what the nitrogenous base is. It's the energy that's in the triphosphate molecule that's going to be used by the individual cell. Now, if you look at the equation I drew here, ATP hydrolyzed to ADP and P releases. If I add those two together, I'm going to get a positive reaction. But I don't really do it with ATP. I do it with UTP. Let's look at it right here. Glucose plus UTP gives me UDP glucose plus fructose 6-phosphate gives me sucrose 6-phosphate plus UDP. Look at the reaction. Okay? Here's fructose 6-phosphate. Here's glucose. Here's UDP. UTP, I remove the phosphate, I take the 2-phosphate, I stick it onto the oxygen on the number 1 carbon. What have I done? I transfer the energy that was in the high energy triple phosphate bond on UTP to the glucose molecule. Now I'm going to allow that glucose molecule, which you see here, to interact with the fructose 6 molecule, etc., and I'm going to form an alpha bond between the two. The overall energy change in the whole process, because the energy of UTP to UDP and ATP to ADP is exactly the same, the overall energy change is minus 1.8 kilocalories per mole. So that reaction is thermodynamically favorable. What did I do? I coupled two reactions together. One that is thermodynamically favored, the one that is not thermodynamically favored to allow it to move. We looked the other day at membrane transport, and I showed you some examples of where you have hydrogen ions going down their concentration gradient, moving things with them in co-transport against their concentration gradient. Same thing, the coupling reaction. That's how you work. Everything metabolically in you combines things that are not favored with things that are favored. So overall, they're favored because you lose energy in the process. Constantly need to supply energy. Here's another example. Glutamic acid, one of the amino acids. Ammonia. I want to stick ammonia on it to make another amino acid, glutamine. Here's glutamic acid. Okay? I want to add to the carboxyl group here another amine group to make the other amino acid. How do I do that? Well, first I phosphorylate the glutamic acid. Then I combine it with the ammonia and I make glutamine. It's a coupled reaction. Overall, the energy change is significant enough to drive the reaction to completion. That's how we work. That's how things go. I'm going to talk about ATP in a little bit of detail. And ultimately what this page is going to do is it's going to lead into how cells make ATP by cellular respiration. It's going to lead to glycolysis, Krebs cycle, and all the others. So I'm going to wait to do that when we do it on Thursday. I'm going to stop now because we've got 10 to 15 minutes. So I might be able to answer some of your questions if you still have questions about the material of the test. So, does anyone have a question? No, everybody's going to get up. The question was, do you need to know by picture what type of microscopy is being shown in the picture? In this case,
Did you send me the 2018 exam?
For example, light uses photons of light.
Did you know
Electron
2018?
microscopy uses
2017?
electrons.
Did you?
Magnets are
You
used
said
to
it.
focus it, glass lenses are used to focus. You need to know the mechanics behind it without knowing which picture is which picture.
I'm looking at it now.
You'll have a lot more.
I'm looking at the exam wall. I think some of the questions are
Over.
The question is, do you need to know the functional groups by picture? Yes. You need to be able to identify, if you see C double bond O dash OH, you need to know that's a hormone. If you see NH2, NH3, you need to know that's an amine. You need to know the functional groups because they have the basic chemistry of those. Macromolecules, you're going to be able to tell the difference between nucleic acids and proteins. I think you should. I'm not going to put a picture of a protein and ask you, is this a nucleic acid or a protein? Estimate the length of a peptide bond. A carbon-carbon bond is this long. A carbon-carbon double bond is this long. A peptide bond is there. It's in between a carbon-carbon single bond and a double bond. It's shorter than a single, longer than a double.
那个什么帮的时候，那个会旋转，会不旋转？
Yeah.
但是它长短没什么。
Yeah.